Melissa. I'm here to present a course on a topic on a course PCT 424, which says stability issues related to semi-solid dosage form. Stability issues related to semi-solid dosage form. The pharmaceutical industry continuously strives to develop dosage form that are effective, safe, and stable throughout their intended shelf life. Semi-solid dosage forms, including creams, ointments, gels, pastes, and suppositories, occupy a crucial niche in drug delivery due to their unique ability to provide localized and systemic therapy. However, these formulations face several stability-related challenges that can compromise their efficacy, safety, and acceptability. Ensuring the stability of semi-solid products is critical for maintaining therapeutic integrity, extending shelf life, and complying with regulatory standards. This study, I want to discuss under it the stability issues associated with semi-solid dosage form, their contributing factor, preventive strategies, and regulatory expectations. Overview of semi-solid dosage form. Semi-solid dosage form are formulations that exhibit characteristics between solid and liquids, allowing them to adhere to surfaces like the skin or mucous membrane without flowing away under gravitational forces. Common types include our normal creams, ointments, gels, pastes, suppositories. Creams, they are emulsions of oil and water, generally intended for topical application. Ointments, they are greasy preparations that provide occlusive effect after used for protective purposes. Gels are semi-rigid system consisting of a network of polymers that entrap a liquid phase. Paste, concentrated dispersion containing a higher proportion of solid material. Suppositories, solid or semi-solid form designed to melt, soften, or dissolve at body temperature inside body cavity. Each of these dosage forms presents unique stability challenges due to their complex composition and interaction with external factors. Concept of stability in pharmaceuticals. Pharmaceutical stability refers to the ability of a drug product to maintain its physical, chemical, microbiological, therapeutic, and toxicological specifications throughout its shelf life. Stability testing ensures that the product will perform a intended under recommended storage condition until the expiry date. For semi-solid dosage form, stability encompasses physical stability. It means retaining its appearance, viscosity, spreadability, and phase consistency while using. Chemical stability ensures active pharmaceutical ingredients and excipients remain intact and effective while stored during or when the customer or patient are using it. Microbial stability, preventing contamination and microbial growth during storage and usage. We have a lot of factors affecting the stability of semi-solid dosage form. The stability of semi-solid dosage form can be compromised by various intrinsic and extrinsic factors. These factors can act individually or synergistically to affect the overall quality and performance of the product. Like the physical stability refers to the product's ability to maintain its original appearance, texture, viscosity, and phase separation behavior throughout its shelf life. There are some, maybe like some creams, if they are not properly produced or developed or compounded in the laboratory. After some time, maybe the shelf life is supposed to take for about maybe 18 months, maybe after 6 months, then you see a kind of separation, the water is going down, some foam is coming up, you will see that there's no compatibility in this particular product. So it is the physical stability referred to the product ability to maintain its original appearance, texture, viscosity, and phase separation behavior throughout the shelf life. Phase separation. Phase separation is a critical problem, particularly for emulsion-based semi-solid like creams. It involves the separation of oil and water phases, resulting in an unacceptable appearance and reduced therapeutic efficacy, like I earlier said. Causes of phase separation: inadequate emulsifier concentration, incompatible excipients, temperature fluctuations, medical stress during handling. Syneresis. Syneresis is a phenomenon where a semi-solid expels liquid, leading to shrinkage and alteration in consistency. It is commonly seen in gels where network collapse over time due to weak structural integrity. Factor promoting syneresis: inappropriate polymer concentration, temperature changes, dehydration during storage, changes in viscosity. That is why after producing your preparation, after doing that, your products, you make sure you maintain the required temperature and proper handling. The packaging material, everything must be put into consideration so that all these things will not affect your product after a time below the required shelf life. Then viscosity is a critical parameter that affects the spreadability and performance of semi-solid products. Loss of viscosity may indicate instability and product degradation. Viscosity changes can result from polymer degradation, phase separation, water evaporation, temperature variation. Then we have chemical stability. Chemical stability involves maintaining chemical integrity and potency of the active pharmaceutical ingredient and necessity over time. Like oxidation, oxidative degradation is a primary cause of instability, especially for APIs and excipients sensitive to oxygen. Unsaturated oil and vitamins like vitamin A and E used in cream and ointment are particularly susceptible. Common causes of oxidation: exposure to atmospheric oxygen, light-induced oxidation, presence of trace metals acting as catalysts. Hydrolysis. Hydrolysis is a chemical reaction involving water that can degrade our active pharmaceutical ingredients, particularly ester, amides, and lactams, leading to loss of efficacy of the products. Factor enhancing hydrolysis: presence of moisture, acidic or basic pH environment, elevated storage temperature, and photo degradation. That is why we must ensure that the pH must not be too acidic and not be too basic as well. We must be in between the perfect line. Preventive measures: use of opaque packaging. If you want to see most of our creams or the gels, like even all the oily gels and the likes, you see that the ones we normally use are most opaque, and if they are not opaque, they will not be as exposed to direct light like this water bottle. There will be a kind of, the color will be deep and light will not be able to penetrate. They will not be transparent. Light will not be able to penetrate directly into the product, thereby affecting the shelf life of the products. So the use of opaque packaging, incorporation of UV absorber, microbial stability. Semi-solid dosage form, especially those containing water, are vulnerable to microbial contamination. Microbial contamination
No,
is a proliferation in semi-solid can lead to product spoilage, potential infections, and reduced efficacy. That is why you see a lot of skin diseases and problems arising in this generation due to all this packaging or in-process development and so on. Source of contamination that can cause microbial contamination: raw materials. It could be from raw material, it could be from manufacturing environment, it could be from improper handling by users, it could be from preservation system failure. That's why when you like creams, after using them, you cover it properly and put in the right temperature. Factor causing preservative failure: adoption of packaging materials, chemical interaction with excipients, inadequate concentration. We have a lot of common stability challenges in various semi-solid. Each type of semi-solid.
Sorry. You say factors that affect preservative failure. What?
Yes, adoption of packaging
adoption
material.
How? I don't understand.
The kind of package material we choose for the particular product can affect the preservation of that particular product, maintaining the shelf life.
I'm seeing adsorption here. Is it adsorption or adoption?
No, adoption to packaging material. The kind of packaging material we adopt to use.
Continue.
Yes, sir. Common stability challenges in various semi-solid forms. Each type of semi-solid dosage form has distinct characteristics and therefore unique stability issue. Understanding these challenges is vital for the formulation of robust and reliable pharmaceutical products, like our creams. Creams are semi-solid emulsion composed of oil phase and a water phase, stabilized by emulsifying agents. They can be either oil in water or water in oil emulsion. Phase separation. Creams are particularly susceptible to phase separation during storage, which can result in a greasy, watery product that is unacceptable for patients. Contributing factors: inefficient emulsification, high shear during manufacturing, thermal cycling during storage, or microbial growth. Due to their high water content, creams are highly prone to microbial contamination. The incorporation of a robust preservative system is very essential. Oxidative degradation. Oil in creams, especially unsaturated fatty acids, are vulnerable to oxidation, leading to rancidity and foul odor. Ointments. Ointments are typically anhydrous preparation intended for topical application and are often more stable than creams. Phase instability. In oleaginous bases like our petrolatum, stability is generally good. However, when incorporating water-soluble drugs, instability may occur due to incompatibility between the hydrophobic base and the hydrophilic drug. Rancidity. Ointment containing natural oil can experience rancidity due to oxidative degradation, especially when exposed to light and air. Hardening or softening. Temperature fluctuation can lead to hardening or softening of ointment, affecting spreadability and patient acceptability. Gels. Gels are semi-solid system consisting of either small inorganic particle or large organic molecules interpenetrated by a liquid. Syneresis. Gels are highly prone to syneresis, which lead to expulsion of liquid and reduction in volume. Microbial contamination. Because many gels are aqueous, they provide an excellent medium for microbial growth if not properly preserved. Polymer degradation in gel. Over time, the gelling agent may degrade, resulting in loss of viscosity and performance. We want to consider paste now. Paste are semi-solid containing a high proportion of insoluble solid dispersed in a fatty or aqueous base. Phase separation. Although pastes are more stable than cream and gels, the solid particle may sediment over time, causing phase separation. Hardening. Water loss due to evaporation can result in a harder, less spreadable product, reducing ease of application on the affected part by the patient. Suppositories. Suppositories are solid at room temperature but melt, soften, or dissolve at body temperature when in the body cavity. Polymorphic changes. Suppositories bases like cocoa butter can undergo polymorphic transformation, leading to changes in melting point, consistency, and drug release profile. Brittleness. Suppositories may become brittle and crack during handling or transportation, especially under low humidity conditions. Microbial growth in suppositories. Water-soluble bases like polyethylene glycol (PEG) can absorb moisture and promote microbial contamination if not properly preserved. That is why, like that suppositories, when it is being dispensed, they will ask the clinician or the dispenser, they ask the patient to put it inside the refrigerator, or if that one is not available, put little water inside a bowl, put it inside prior to usage time. Strategies to enhance stability. Addressing the stability challenges in semi-solid dosage form requires a multidisciplinary approach involving careful selection of formulation components, optimized manufacturing processes, suitable packaging, and appropriate storage condition. Formulation strategies. Selection of stable excipients have a lot to do in the products and the shelf life of our semi-solid dosage form. Choosing excipients that are chemically compatible with the active pharmaceutical ingredients and with each other is crucial. Antioxidants like butylated hydroxytoluene or butylated hydroxyanisole are often included to prevent oxidative degradation in semi-solid dosage form. Use of emulsifiers and stabilizers. In emulsions-based semi-solid like creams, emulsifiers and stabilizers help maintain phase stability. Surfactants such as polysorbates are commonly used to enhance emulsion stability as well. Incorporation of preservative. Preservatives such as paraben, benzyl alcohol, or phenolxyl ethanol are vital for preventing microbial contamination in water-based semi-solid formulation. Factor to consider in preservative selection, because all these excipients have a lot to do. Even when you have a good API, being very careful in the kind of excipient you select in your preparation and drug development is very, very crucial. So it will bring a positive and a good product to the society. Spectrum of activity must be taken note. Compatibility with formulation components also is very, very crucial. Stability under storage condition is very, very crucial as well. pH adjustment also. Maintaining an optimal pH can minimize hydrolysis and microbial growth. For example, buffering agents may be added to keep the formulation within a stable pH range. Process optimization. Controlled homogenization. Homogenization parameters like speed, time, temperature must be carefully controlled to ensure proper particle size distribution and emulsion stability without degrading sensitive ingredients. Temperature control during manufacturing. Maintaining appropriate temperatures during the mixing and filling process prevents degradation and phase instability. Packaging strategies. Use of protective packaging. Packaging that limits exposure to light, air, and moisture enhance stability because it will prevent antimicrobial or air from entering into the products. For instance, aluminum tubes, airless pumps, and opaque container are frequently used for semi-solid most often. Material compatibility. The interaction between the formulation and the packaging material must be evaluated. You must be very sure that the packaging material you are adopting must not be the one that will react with your product. Reacting with the product will break down. You will not be able to maintain the shelf life you have proposed for it. So single-use packaging. For products prone to microbial contamination, like all these our ophthalmic gels, single-dose packaging can significantly enhance microbiological stability. Storage and handling. Proper storage conditions are vital for maintaining stability. Recommended strategies include temperature control. That's why you see, there's no how you enter into any pharmacy store, you will see their pH regulator and their thermometer, just to make sure that the temperature of that of the store is maintained. Temperature control. Most semi-solid products are stored at room temperature, 15 to 25 degrees centigrade, unless otherwise specified. Protection from light. Storage in dark environment or opaque packaging prevents photo degradation. Avoidance of repeated free store cycle prevents phase separation and texture change in emulsion and gels. Educating patients on appropriate storage and usage practices is equally important to preserve product integrity after dispensing, so that we can optimize the treatment, the treatment focus on the patient, because it's a thing to give the patient a particular drug in good condition. It's another thing for the patient to be able to maintain the storage throughout the period of the treatment. Regulatory aspects related to stability. Regulatory agencies such as the FDA, the Food Drug Administration, World Health Organization, and International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, ICH, provide specific guidelines to ensure the stability of pharmaceutical products, including semi-solid dosage form. That is why you see them, they visit all those production companies to make sure they adhere to the regulation given to each production and manufacturing. Stability testing guidelines, ICH guideline. The International Council for Harmonization has published a series of guidelines, e.g. ICHQ1A, R2, detailing the requirements for stability testing for new drug substances and products. Key aspects include testing condition, long-term, intermediate, and accelerated stability study under specific temperature and humidity conditions. Testing parameters: physical appearance, viscosity, chemical acid degradation product, microbiological preservative efficacy, microbial limit test. Shelf life determination based on the time during which the product remain within specification limits under recommended storage condition. WHO guideline. The World Health Organization emphasizes the need for stability testing for pharmaceutical products, especially for those intended for global markets with diverse climatic conditions. WHO classifies stability zone based on temperature and humidity. Zone 1, temperate. Zone 2, subtropical and Mediterranean. Zone 3, hot and dry. Zone 4, hot and humid. Semi-solid dosage form intended for tropical countries must demonstrate stability under harsh conditions. This is just telling us that the area you are formulating a particular drug for will determine the kind of the condition you are going to put in place while formulating the products. Like when you are going to the north, the cream you want to produce for maybe the west customer, you should know that over here we don't have that harsh weather compared to those in north So by the time you are going to transport those drugs, even on the way during logistic, everything is just destroyed by temperature, hot temperature and the like. So all these things are to put into consideration the kind of zone you are working with when producing your drug. Federal Drug and Food and Drug Agency guidelines. The US Food and Drug Agency requires comprehensive stability studies during the development and approval of semi-solid products. Important aspects include first degradation studies to identify degradation pathways, evaluation of container closure integrity, demonstration of microbiological stability, particularly for multi-use products. Stability indicating method. A stability indicating method is an analytical procedure that accurately and precisely measure active ingredients without interference from degradation products, impurities, excipients, or other potential sources. Common analytical methods that we use include high performance liquid chromatography, gas chromatography, spectrophotometric assays, microbial limit tests. All these methods are validated according to International Council for Harmonization guidelines on the validation of analytical procedure. Documentation and shelf life assignment. All stability testing data must be compiled into a regulatory submission dossier, including the protocols, the testing results, statistical analysis, proposed storage condition and shelf life. For semi-solid dosage form, real-time and accelerated stability data help establish an appropriate expiry date and storage recommendation. Case study of practical examples. Examining real-world example of stability issue and solution in semi-solid dosage form provide practical insights into the challenges faced during formulation, development, and commercial production. Number 1, stability of a hydrocortisone cream. A study investigated the stability of hydrocortisone cream formulated as an oil in water emulsion. Observation. Phase separation occurred after 3 months at 40 degrees centigrade, 75% accelerated condition. Loss of drug potency by 8% due to oxidative degradation. Solution. Incorporation of antioxidant, e.g. BHT, minimize oxidation. Use of a more robust emulsifier system stabilize the emulsion against thermal stress. Packaging in aluminum tubes reduce oxygen permeability, enhancing shelf life. Case study 2. Microbial contamination in a topical gel. An antimicrobial gel intended for wound healing showed microbial growth after 6 months of storage under high humidity condition. Observation. Failure of the preservative system due to interaction with jelly agent, e.g. capable. Solution. Replacing paraben with a more compatible preservative like venosyl ethanol. Adjusting pH to optimize preservative efficacy. Implementing aseptic manufacturing practices reduce initial microbial load. Case study 3. Stability changes with suppositories. Suppositories containing diazepam formulated with cocoa butter experience polymorphic transition leading to cracking, non-uniform drug release, melting point variability. Solution. Using synthetic base like white salt, which have more stable polymorphic property. Strict temperature control during production and storage. In conclusion, the stability of semi-solid dosage form is a critical factor determining therapeutic efficacy, safety, and patient acceptability. Physical instability such as phase separation, viscosity change, and synergies syneresis, as well as chemical degradation like oxidation and hydrolysis, are common challenges that must be addressed during formulation development. Microbial contamination is another significant concern, particularly for water-containing semi-solid like creams and gels. Though, through the careful selection of excipients, incorporation of stabilizer, use of appropriate preservative, optimization of manufacturing process, and thoughtful packaging choices, formulators can enhance the stability profile of semi-solid products. Regulatory guidelines provided by organizations such as International Council for Harmonization, Food and Drug Agency, and World Health Organization set the standard for stability testing, ensuring that semi-solid pharmaceutical products maintain their intended quality throughout their shelf life. Real-world case study further demonstrates the complexity involved, the necessity of systematic approach to formulation and stability assessment. Ultimately, a thorough understanding of stability principles leads to the development of safe, effective, and reliable semi-solid dosage forms for patient use. Thank you, sir.
Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much for the wonderful presentation. That was a detailed work. All right. Thank you very much. God bless you.
Thank you, sir.